how you would set up the interference panel, and then how you would compare that to your limit of blank and limit of detection.
Hey Nick, we've been speaking about clinical trials and clinical trials as a product of product development.
Yeah.
So can you tell me a little bit more? Let's say I'm a developer and I'm approaching this moment where I need to run a clinical trial. Like, where do I even start?
Yeah, that's a great question. I think the last time we talked about different phases of clinical trials, and I don't think we got into like how you actually do that.
Mm-hmm.
And look, there's experience, of course. We do this quite a bit, but really, what I look to is the Clinical Laboratory Standards Institute. They have a whole series of guidance documents that really talks about every aspect of designing clinical trials, right from power calculations to see how many subjects you need in, to designing the actual trials. And so that's what I go to. I think the main document that I often look at is CLSI EP 17A2, and that is the guidance that's specific around how you determine the limit of blank, your limit of detection, and most importantly, our old friend, limit of quantification.
There you go. So let's say if I was chewing gum,
Sure.
Is there any guidance on how to deal with like substances that you may encounter in the environment?
Oh, sure, like when you say chewing gum.
Substances?
Yeah, that's a great question. And in fact, CLSI has it covered there as well. It's guidance document EP7A2 that covers interfering compounds. And when you're developing COVID diagnostics, you have the saliva test or the deep nasal test, they cover all the different interfering substances that you would find along the way, particularly in saliva, things like caffeine and other things that would interfere there. They talk about how you would set up the interference panel, and then how you would compare that to your limit of blank and limit of detection.
There you go. And then you're speaking about limit of detection and limit of quantification. Right. In terms of precision and bias, are there documents for that?
Yeah, top of the class once again, Joris. Yes, precision and bias. Our friends at CLSI have us covered yet again. The document there is EP 15A2, I believe, and that reference guidance explains how you do all your calculations and set up to account for exactly that, precision and bias.
There you go. So it sounds like these documents have you covered when you want to start a clinical
CLSI
trial.
is the reference for how you go about setting up these trials.
There you
Absolutely.
go. Thanks very much, Nick. That's very interesting.
You're welcome.
You have a great day.
Have a great day.